Thank you so much, Rachel. From the background survey, I saw lots of people have actually analytical background. So I'm thrilled and also a bit like, feels that I might have, tempered down my slides too much. Probably should have included a bit more mathematics.
No, I think it's fine. I think it's fine, honestly.
Okay. But yeah, thank you so much, guys, and thank you, Richard, for the opportunity. Really delighted to be here. So Kat and myself, we are both working within this organization, which is part of the NIHR funded infrastructure called NIHR London In Vitro Diagnostic Cooperative based at Imperial College. Today, we're going to do a short talk telling you guys about multi-criterial decision analysis and how we use it in our unit to help assessing health technology. Actually, today's case study, we focus on a biomarker development. Thank you, Kat. Yeah, so we will say briefly who we are and what led us to use the multi-criteria decision analysis and why do we need it in innovation, which is what we do on a daily basis. And then we'll present case study of MCDA, but it's a particular format of MCDA, multi-criteria decision analysis called SMART. So SMART is a really simple execution of MCDA. Then we will basically tell the story of how we use the MCDA or SMART approach to help to accelerate the biomarker development and translation. So this is me, and more information available on our website. So I have a background in digital analysis, having worked with some really proper experts in the field from London School of Economics, where I did my PhD. So currently within our unit, I work with people who are developing technologies from the very early all the way to the very late stage, including accessing the market. So we perform all sorts of analyses, including human factor, cost, health economics, stakeholder analysis, and so on and so forth. So Kat, would you like to say who you are?
Yeah, sure. Thanks from my side as well for the opportunity. It's great to be here and talk to you. So I'm Katerina. I have a background in biomedical sciences, and then I did my PhD in biomarker translational research. Currently, I'm part of London In Vitro Diagnostic Cooperative at Imperial College, and I'm working on translational research as well as cost evaluation of diagnostics mostly and other innovations.
Thank you, Kat. Yeah, so we support innovator based on innovation stage from ideation all the way to the market. So this is basically the developmental pathway. So within London IVD, what we do is that we generate what we call the multi-dimensional evidence in terms of, qualitative evidence, quantitative evidence, quantitative evidence such as health economics, qualitative evidence such as those extracted from interviewing focus groups and in basically understanding who is going to use a device, in what context is that device going to be used, and for whom the benefit and the risks might be maximized, the balance might be addressed. And so we use the multi-dimensional evidence to work with innovator, to work with healthcare systems to support commercialization and to make sure that the evidence is used objectively and appropriately. Thank you, Kat. So what is multi-criteria decision analysis? What is MCDA and why do we need it? So the irony is that in our line of work, we often encounter technologies already being developed to a fairly advanced stage, but not clear, what its unmet needs are and how do they plan to meet it. And what is interesting is that such technology, or they might have locked in the lab for 10 years, right? And then they suddenly being thrown out of the lab into the real world, and then suddenly it's almost like not adapted to the real world and the freak out. Say, oh gosh, yeah, I have this great idea, but nobody's going to use it. What I'm going to do? So we believe that if we engage these, developer early enough in the developmental pathway, such as the concept stage, we can help them use the evidence to properly shape the idea, including, the cost effectiveness, including the design, including who they need to engage with during the developmental process. So not just lock themselves up in the laboratory, but really to understand the consequence of technology and what needs to be done. So do the proper planning, but use evidence to support that journey. Next slide, please. So challenges of innovative decision makers often include, like, we have developed a device, but nobody wants to use it. We have a limited budget of time from decision maker point of view. So which project shall we support? And we got a great idea to work on, but how do we make sure it's value for money? And we have many design options to choose from, which one to take, etc., etc. We believe that these challenges actually reflect the complexity of the healthcare ecosystem. And in particular, I mean, along the, as you guys can see, along the developmental pathway, there are many different stages, there are many different stakeholders, and they have different perspective and preference. The challenge is how can we actually capture those perspectives and preferences, converting them into evidence that the developers can use to inform the product development. So it's almost like a translation of people's preference and perspectives into usable evidence. And so the complex decision always have many options and objectives, and there are uncertainty that cannot be reducible. Nobody anticipated COVID-19, for instance. Nobody anticipated, we have to, under extreme time pressure, we have to accelerate the take up of device and medication such a short time turnaround. And most of all, we have only limited resources, including money and time, to allocate to a project, to development, or to the market. And some multi-criteria decision analysis is basically a socio-technical solution to next slide, please, Scott, to the challenges we believe. The social, in terms of there are so many different stakeholders, and we do need to leverage on, these people early on enough in the innovation pathway. Technical, in terms of, there is theories and the models that we can utilize that actually is mathematically sound. So there are rational decision-making theory that we can use to support this discussion. So these two are the founding father of the multi-criteria decision analysis approach, and Kinney and Reifer. So this is a classic book which kick off the entire field of the MCDA. So it originated in America. Then my mentor at the London School of Economics, Professor Larry Phillips, is one of the first persons to bring the multi-criteria decision analysis to the UK and have the decision science degree, which I got as part of my postgraduate study. So Larry Phillips, he invented something called the decision conferencing process to enable the MCDA, because it is a social and technical. And the social, in terms of, we need to identify a group of key players. And in our lingo, we call them stakeholders. So these people will have a stake in the technology. They might be impacted by the technology, or they might actually have an influence on what technology can be developed. So these people are scattered around in the healthcare system, in addition to people like who develop technology, in addition to patients, in addition to users, such as clinicians, such as, the managers and people who pay for it, like the commissioner. All these people are stakeholder, and their role cannot be ignored. And increasingly, patients' perspective are very important. So Larry Phillips had this idea of putting people together in a room, but in a room in a quite specific way. So people have to be able to see each other. The group size cannot be too large. to really leverage on the social component of this process to enable a fruitful discussion. And the model, he argued that the model is best developed on the spot. So we don't have a preset agenda. Apart from we know what are the problems we're going to working on and what we want to achieve. So objective and challenge, but not the specific solution to it. He believed that decision conferencing supported by a facilitator who is an expert in social process, such as himself, can develop the model on the spot and then use the model to explore the issue, to explore the solution, and then we can create a shared understanding of the problem and together develop a commitment to the ways forward. And that model basically is the MCDA model. Next slide, please. So the MCDA, there are many different forms of it. And today, just going to introduce you guys a quite simple one called the SMART process. Simple in terms of, mainly in terms of how, the number can be aggregated in a very simple way to aggregation. So the steps including defining the problem, we call that process problem structuring. It has been argued that the problem structuring it needs to take really proper time and effort to really understand what the issue, and that is probably one of the hardest things to achieve. So what is exactly the question that we are going to ask, and that is essential that we have the right people in the team. So the stakeholder identification already taken place at that stage. Then we develop a possible list of solutions to the problem. And from the list of solutions, we understand what are the criteria can help us identify which solution might be better. Then we score the solution against those criteria, coming up with something like a decision matrix, whereby we group those information. However, not all criteria are equal. For instance, if we trade off cost and life, right? People will, of course, argue that money is not as valuable as saving life. So there is a trade-off between different criteria we need to consider. And then we convert, after we have converted both the scores against each criteria and the relative importance of those criteria into numbers, we can synthesize them into an overall score, and looking at the performance of the alternative on those criteria and the overall scores, and to generate the solution, which is decision-making process. Next slide, please. So I'm just going to give an example of the biomarker, which Kat will also follow up on my talk. So basically, one frequent issue we encounter in health technology is that the use of biomarker. So here is an example of acute appendicitis. We published the paper 2017 in surgical endoscopy. So the challenge is that acute appendicitis is the most common surgical emergency procedure. However, negative appendicectomy rate is as high as 20%, which means that 1 in 5 people, when you believe they have acute appendicitis, but in reality they don't. So it's a huge impact on the patient's life, as well as the cost to the NHS. So the inspiration is that, can we improve diagnosis better? And there are a variety of biomarkers can be used to achieve that. An ideal biomarker simultaneously can maximize clinical utility, but minimize costs as well as time, because time here is crucial. Next slide, please. So what we did is that we look at the literature with systematic search and look at potential biomarker. And we also did a survey of the consultant surgeon to understand, what they value when they select those biomarker. So from this process, we extracted 6 biomarker, non-invasive biomarker, that potentially could be clinically relevant and easily translatable, and 8 relevant performance criteria. So the next slide will show you what these 6 biomarker and 8 performance criteria are. So white blood cell count, CRP, bilirubin procalcitonin, and the urinary serotonin, and interleukin-6. So in the table on the each column is one of the 8 performance criteria those consultant surgeons as well as literature tell us are important, and we have extracted some number and did the synthesis using meta-analysis. So for instance, the first row, white blood cell count, sensitivity diagnosing acute appendicitis is 79%, specificity is quite low, 55%, but there are also other attributes that the people believe are important. So how easy it is to do the test, and how predictive it is of the perforation risk of a patient having appendicitis, how much it costs, how long it takes to generate the results, how acceptable it is to the clinician and the patient, how reproducible the results might be. The reason I have these 2 red box blocked, it will become clear to you later. At the bottom of this table is a surgeon rank. So they rank the sensitivity is the most important, which is not surprising given our main goal is to rule out the false negatives. The high sensitivity, the more likely we will have a correct identification of appendicitis. Next slide, please. So we got this information, but to apply the district analysis, we need to score the option. And score the option meaning that we convert those actual sensitivity and the specificity and so on and so forth into the score. And in the MCDA jargon, it is something called the interval scale. So we basically is a 0 to 100 scale. 0 means that the worst option. It doesn't mean that it has no value. It just means that in the context of the decision problem, and of the options, 6 options that we are looking at, 6 biomarker, one of them is the worst on this particular criteria, and the other is the best. So if we use sensitivity as an example, the highest one is the white blood cell count. So it is scored as, there is some animation here, Kat, if you could just, yeah. So the highest one is scored 100, the lowest one is scored 0. So procalcitonin and the white blood cell count, they assign the score of 0 and 100 because they have respectively the lowest and highest sensitivity, and thereby define the scale of the sensitivity scoring. Then we basically convert the remaining into the relevant, but use a very simple linear transformation proportional to the scale defined by these 2. Next slide, please. And in the weighting, not all criteria created equal. So here, what needs to be noted is that the weighting reflects both the range of difference and how much that difference matters. And so we ask, is the difference between 0 to 100 on one preference scale, such as sensitivity, how does that compare to the same difference from 0 to 100 compared to another scale, such as specificity? So that comes up with the swing weight. Next slide, please. And so now the reason that I block these 2 becomes clear, because although ease of test and acceptability are important, and the surgeon does rank the ease of test, for instance, actually as number 3 most important criteria. However, all the options they actually score, they rank the same as ease of test, which means that they have no swing because the difference is 0. So these 2 actually are removed from the analysis. Next slide, please. So once we got the weighting using the swing weight procedure, we normalize them. And after we normalize the swing weight, and because we already at that stage score the options on each of the 6 remaining criteria, which they differentiate, we can put the scores together into something between the 0 and the 100, with 100 as the highest score. And in the table, basically, is the results of this analysis. So the cost performance, time performance, and the diagnostic benefit. The diagnostic benefit is a synthesis of the sensitivity, specificity, reproducibility, and the predictive power of perforation. So for instance, at the first row, the cost performance CRP is the worst because it's most expensive. Bilirubin is the best 100. It is the least expensive. And in terms of diagnostic benefit, the urinary serotonin, which is the last one, to the rightmost column, scored the highest, whereby the CRP actually scored the lowest in combination of the sensitivity, specificity, reproducibility, and perforation. So of course, as we do this analysis, there are lots of uncertainty. So we before we say, okay, we're going to select one or the other. Next slide, please. We need to look at uncertainty. And what we do is that we look at the uncertainty many different ways. This is just one way of looking at it. And so this is looking at how the benefit relative cost and how the benefit relative to time in terms of the weighting we assign to them. And as we can see, for instance, the one that have the overall high score, which is bilirubin, and the second best option, which is the white blood cell count, represented respectively by the green and the black line on the graph. They're actually really close, and also they're very sensitive to the weight. So the current weight is indicated by the dashed line. So basically, cost is 80%. Cost and time are up around 80% actually to the benefit. And you can see that with a slight change in the dashed line, imagine that the graph of the benefit relative costs. So imagine that line, dashed line, moves slightly to the left, that it will result to a difference in which option is the best. Currently, I think the option the best is the interleukin-6, but by moving slightly to the left-hand side, reducing the importance of benefit relative to cost, the best option or the ranking of the the overall ranking of the benefit score will change. So the results of this analysis by no means actually robust, which means that although we come up with some number, and when we generate the decision-making stage and the ways forward, when we do this analysis, ideally on the spot with a group of stakeholders and decision maker, sitting together, we can have a proper discussion about what we truly value and what evidence that we need next step in order to make our conclusion more certain. Next slide, please. So this is my last slide before I hand over to my colleague, Kat. So from this research, what we think and from the reaction we got by working with the people, the surgeons, the clinicians, the patients, we think that MCDA really provide a very good structure for thinking through a complex problem. And so we believe that, especially decision matrix, once we understand the criteria on which we can evaluate the option, actually we can use that as a leverage to think more broadly about what the biomarker development. Imagine that at a very early stage, if people know that these are the important criteria, like for instance, I want to develop a biomarker for diagnosing cancer. Imagine at the beginning, I already know that to diagnosing cancer, these are the important things I need to consider. So from developer perspective, they know that what they need to focusing on. So it's not just like isolate themselves in the lab and trying to maximize sensitivity, specificity without considering how much it costs, how much it is time to produce. So they can have better chance of being successful in the market. So that inspires Kat's research. Kat, can I hand over to you, please?
Thank you. So the next few slides will focus on a decision matrix project that we did as part of my PhD, which was supported by Chris Peters, Melody, and also Professor Hannah. So the main project that we're trying to develop is the biomarker toolkit, a tool to mediate the successful translation of biomarkers from lab to clinic. To start with, I wanted to give you some background regarding biomarkers. So this is a biomarker translation pipeline, which shows the basic steps that result to the translation of a biomarker, starting from discovery, moving on to assay validation, clinical validation, and lastly, clinical application. Even though it seems very simple, over 1,000 of breast cancer biomarkers are identified, but only few are currently clinically used, which shows the importance of developing this tool that will help us decide which biomarkers are more promising ones and could move along the pipeline. So the ultimate impact of the project is to target biomarker research, reduce the cost associated with excessive discovery, utilize time more effectively, and therefore result in more efficient biomarker clinical implementation. So to start and develop the biomarker toolkit, we have two main aims. The first one is to develop the backbone of the biomarker toolkit by identifying which characteristics make a clinically useful biomarker and converting this into a checklist. So to develop the checklist, a systematic literature search was conducted with literature and guidelines as well, public and patient involvement, semi-structured interviews of experts in the field, for example, from people in industry, clinicians, and academics as well, and a two-stage Delphi survey with biomarker experts. So upon development of the checklist, it was then validated using breast and colorectal cancer. And in the last stage, we aim to assess the usability of the toolkit. So in the next few slides, I'm going to go through briefly the two main stages of the biomarker toolkit. So as I said before, systematic search was conducted within the other methodologies to identify what makes a clinically useful biomarker. Upon completion of all of the stages, 129 characteristics were identified, which were grouped into four main categories, including rationale, which covers mostly aspects of burden of a disease and why are we deciding to choose and select a specific biomarker. Analytical validity, which covers aspects of experimental design, biospecimen quality, and quality assurance. Clinical validity, where aspects of population selection, characterization, the use of specific statistical analysis, and sensitivity and specificity of a specific marker are included. And lastly, clinical utility, which involves aspects of cost effectiveness, feasibility, human factor, as well as regulatory authority approval. So all of these characteristics formulated the backbone of the biomarker toolkit, which was then validated using successfully translated biomarkers and stalled biomarkers. By successful biomarkers, we mean that they're clinically used, and stalled biomarkers are currently not clinically implemented. We chose breast and colorectal cancer biomarkers to start with our validation because in these cancer types there are high level of research with biomarkers which are actually clinically useful. Therefore, we could test our checklist and assess the toolkit. Just to make you understand how does the toolkit work, first of all, we conduct systematic literature search for each biomarker that we select. Then we identify relevant articles. We read them and then score them based on the presence of the characteristics that I have previously described. Then using in-house algorithms, we calculate scores for each of the biomarker, and then we compare them between the stalled and the successful group. So these are the results that we got from the successful biomarkers and stalled biomarkers in breast cancer. And as you can see, there is a significant difference between the two groups. While a difference is also seen in individual categories, with the biggest difference seen under clinical utility, which covers aspects of cost effectiveness and human factors, as well as feasibility. And interestingly, the same pattern was observed in colorectal cancer biomarkers. So a higher score was scored in successful biomarkers. And this higher score indicates that the specific publications of successful biomarkers cover most of the aspects, more aspects of the biomarker toolkit checklist in comparison to the stalled group. And once again, differences are reflected in individual categories, with the biggest difference also seen in clinical utility. This suggests that the biomarker toolkit is very promising and can be used to indicate which biomarkers are more successful are more likely to succeed, basically. In this way, funders, clinicians, and academics can be guided towards biomarkers which are more likely to be clinically implemented, while also this tool can be used to guide a biomarker researcher to identify any gaps, for example, in specific categories in their research, in order to promote biomarker translation and prevent them from being stalled. So currently, the biomarker toolkit requires human input and human effort, as we want to read and score the papers manually. However, future work aims to automate this by using natural language processing. So we want to automatically identify the articles for each biomarker and then score them based on the presence of specific characteristics that will be automatically extracted. This way, it will improve the usability of the tool and also improve its effectiveness and applicability by the end users. So our final vision is to have an application with different characteristics inserted into it. And these characteristics will generate scores for the different categories at different stages of biomarker development. And each score will reflect the ability of a biomarker to be clinically used and can also be used to identify any possible gaps during research in order to be able to promote a trajectory of biomarker development that will assist in their clinical implementation. So that was a brief summary of the tool, which also overlaps with what Melody discussed before about deciding basically which of the cohort of biomarker, of the pool of biomarker, is more likely to succeed based on scores. It's a different kind of scoring, but the concept is very overlapping.
Thank you, Kat.
Thank you.
So we have 2 more slides. So we believe that multi-criterial analysis, even just as a thought process, is a very useful framework for thinking about and generating solutions about complex problems in the complex socio-technical systems, which is very characteristic of this, NHS and biotech, medtech landscape. The success ingredient is really we need to identify the key player, engage and involve the stakeholder, of course, including patients as well, and as well as robust evidence. So evidence can be used to reduce uncertainty, and the model can be used, the MCDA model can be used to capture the evidence. And here I want to say that the evidence doesn't limit it to only evidence, the published evidence. Evidence can be expert judgments, can be, of course, can be generated by randomized control trial. But very often in decision problem, especially complex ones, that such evidence might not be available. So we often have to talk to people, get the evidence like expert judgments and other kind of study to incorporate that. And very important that we probably didn't mention this is that the clinical pathway evidence is extremely important. That is what CAT captured under the implementation component. And the clinical pathway very often can only come from the interviewing people. So it is really the process that matters, because it's not just that we want to generate solution, we also want to generate interest and the commitment towards the ways forward. We want people to understand, how the process works. We don't want to give them a black box, just say, okay, this is the other option, this is the best option using MCDA. I think that really defeats the purpose. We want them to understand how the score, for instance, biomarker score is being generated, and why we think this biomarker might be better. Therefore, in the CATS tool, she didn't go into it with limited time, but we're very happy to tell you more in another talk or study, is that with the biomarker tool, we want to give them feedback, based on the score, might be, the ways forward, the evidence can be generated to improve the scores, and so that, the biomarker development might be more likely to success. So it's really about working with the stakeholder, generate shared understanding, and co-create ways forward. So the last slide is some reading, and there are plenty, but just for our talk today is the 2 paper we published using the multi-criteria digital analysis approach, and there is this at the bottom is a community of and the local government. This is really like available freely online, a manual, very easy to read manual. A bit long, unfortunately, but it basically is a step-to-step guide how you can actually execute the MCDA process. Thank you, everyone. Yeah, feel free to get in touch with any questions, and our unit website available. Thank you.
Thank you, everyone.
Thank you, Melody. Thank you, Kat. That was fascinating. Thank you for that intro to MCDA for those who wanted to know a bit more about it and some detail about how you've used it. So I have to refer back to the 10th anniversary of the strategy unit party yesterday, which wasn't, it was an away day, but we did actually. When they made us think hard, they presented us with certain scenarios, and what would you do in this type of scenario? And one of them was constructed by Stephen Wyatt, who isn't here today, so I can say whatever I like. This was about the proposed closure of a hospital and the damming of the hospital for its health and safety sort of situation and so on. So all these things put into this scenario, lots of stakeholders, very complex. And we were saying yesterday, apart from the fact that it was into the afternoon and we were tired and we didn't want to think anymore, that MCDA, from those who had some introduction to it, that'd be ideal for that kind of situation, which is very different from sort of looking at biomarkers and something quite specific. But equally, it's having that process in place, the importance of having the process and the stakeholders and everything, but having that robust approach to deciding collectively with those stakeholders what the criteria is, and going through that process of weighting them and then agreeing them, and then the decisions are, you would hope, is easier to make. But no, that was I really enjoyed that. That was fascinating. And thank you also for sharing the resources that you shared at the end, albeit one of them's long. I'm sure people will definitely sort of pursue that. We had somebody has said I forgot to say various things actually. I forgot to, and one of them was if people do have questions, for them to put them in the chat or onto the Menti questionnaire, because what happens in these, about 50% of people can access the chat. I don't know if you can, Melody and Kat, and 50% can't. Yeah, so some can't. So there is a question in the chat. So before we go to Mohammed's point, I just want to say on Menti, somebody who's obviously been here before and knows that is always something that exists for people to interact with me and presenters. They say it's amazing work. There's a long journey ahead in our organisation to go from the descriptive analysis we currently do to this far more advanced type of work. So it's taking it's those early stages, isn't it, when you do that descriptive analysis, having in mind how that's going to be used, and sort of bringing into that at those very early sort of conceptual stages almost, that end point that you want to get to in order for it to be successful. So what does Mohammed says, can you say more about how long the process takes, and how many interactions with stakeholders are usual? So I'm sure it varies, but would you like to respond to that?
Yeah, I will say a few words, and I'll hand over to Katrina. Katrina will say that's 3 years. That's how long her PhD lasts. But in response to that, I think that's one reason that Larry Phillips created the decision conferencing, because decision conferencing can probably solve a problem in the space of 3 days. So, you know, 1/2 day spent in discussing the problem, and the 1 day spent in, you know, developing the model. Then 1 final day spent in generating solution, looking at the uncertainty, agreeing on ways forward. So it can be quite short process. However, you know, what we realize is that that model is probably more applicable to when you engage in a very policy-making perspective, like decision conference. You need a very experienced facilitator. So what we do is that we do a different version of it. So we do stakeholder engagement throughout our research. And Kat, would you like to say a bit more on that?
Yeah, I think you covered most of the question. Just about the stakeholders, I would say usually we engage with our contacts, because it's very difficult to find someone who is in charge for the specific topic that you're looking at. But as a rule of like the analysis, we continue recruiting people until we get saturation. So when we analyze the themes and they all agree with the pathway that we have later with the topic. So I would say you keep on recruiting as many people as you have for the specific field until you have no more new data.
Thank you very much. So Mohammed, did that answer your question? If you don't say anything else, say yes, it did. Because so from my limited understanding, we have a hand up actually. My limited understanding. So while I'll come to Jack in a second. My limited understanding. We had somebody who came and did a session with us, which was called Rigs to Reefs. Was it that way around? It was all about MCDA, and it was all about using the I'll tell you what, I'm going to put a link in the chat, because otherwise I'm going to use up all the time talking about something which happened in the past. I want to get to Jack in a second or two, but from my sort of what I remember from that is that huge importance of the process, that the decision, the conference, and getting the right people around the table at that time, and that can be relatively quick, but the preparation, the work behind it, the build up is all extremely important for that to be quick, the preparation, the planning. Jack, do you have a point? Question?
Yes. Hiya. Hello, thank you, Rachel. I thought that on the Rigs to Reef talk, what I thought was really fascinating is they said that often you don't need to finish the whole analysis to get a decision, but halfway through the decision might become clear of what you need to make. And I think it was all through that they also the decision was created through the process. It wasn't like they had a series of options at the beginning. They sort of come up with it through it, which I thought was fascinating. But anyway, sorry. No, it's great to. Yes. I'm a health economist working in the HEU. We've been using a socio-technical approach for the last year to help integrated care systems. So it's been really great to hear your talk. I have 2 questions. The first is, I know that NICE were considering MCDA as part of their formal HTA process, but that's all I know. Do you know more about that?
No.
Firstly,
Unfortunately,
no, okay.
I don't have insight into that. Yeah, I would love to work with NICE too.
Yeah. Well, so would I. And my second question is a specific one. So we've got a problem at the moment where there's a need for us to engage with more stakeholders than would be reasonable to get in a workshop decision conference. And one thing we're thinking through is whether we can use some form of survey to do direct rating for the scores. However, I haven't found any examples of that in the literature. I know surveys have been done to attribute value judgments to quantitative scores, but not to do direct rating. I wonder if you had any thoughts on that or any examples of where that's been done before.
Direct rating, you mean that using survey just asking people to directly rate an option against a criteria? So you can do that, but you need to define the criteria and what do you mean by rating very clearly, because I mean in the SMART, for instance, it's something called the interval scale. So you need to explain to people, you know, what 100 means, what 0 means, and that definition is crucial, right? So people need to really understand that definition. So the communication, rather than, you know, you can communicate everything on the spot so people can ask you a question, you would be expecting that when they see the survey question, they understand automatically what you mean, and everybody has quite similar. So that is the biggest caveat when do the survey. So the same understanding and the same methodology, and people give you the number actually reflect what you want. That kind of validity, yeah, in translating your, you know, language into that, then translate back. Yeah.
That makes sense, and I guess it's much more easier to work out if people understand what you're doing if
Absolutely.
it's in a workshop.
And test it as well. Yeah.
Thank you. That's really helpful.
No worries. Yeah, feel free to email us any further question. Yeah.
You may regret offering that.
No, never.
Jack, don't be a bully. Okay, well, that's great. I think so. We don't have any more questions that I can see. You've shared your contact details, and I can share those after the session as well. So I just want to say another thank you to both of you for coming today and sharing your work. I'm sure it's been an inspiration to many of us, and I think it's this sort of language, these types of techniques are becoming more I wouldn't say certainly common, but more people are hearing about them and about the utility of them. So thanks again for sharing the example that you did today, both of you. So I'm just going to I've got a couple of minutes just to share a couple of things that I would like to with. Oh, can I just do this again? I know this is probably really annoying, but I'm going to do it. I like my toys. Yeah, mentioned it already, of course. So this is the Health and Care Analytics Conference. A few people on the call today. I think I've seen one or two who are going to be presenting there, online only at the moment because face-to-face is sold out. But don't worry, because we're going to do it again next year and the year after that. So the full program is going to be out probably next week, or certainly a lot of info about the full program you've all been waiting for. So do register for the online experience. And coming up in the next few huddles, we've got Dan Isaac. He's going to be doing a taster session for his data visualization one-day training course, which is in about, the course starts in about, here comes the dog. Hello, good boy. About 3 weeks, maybe about 4 weeks from now, so to look out for that. And Stephen Wyatt's going to be talking to us about hip replacement equity trends. He's been doing the rounds with some of that info, that talk at the moment, and it's very popular. I do recommend you come to that one. And we're going to be hearing more about the data science team in the strategy unit, and the NHSE are presenting the ICB place-based allocation tool. So these are the ones which are coming up in the next couple of months, but there is more. And of course, if anybody wants to come and share their work, they are very, very welcome to do that. So just mention the training opportunities, go to the website, and very finally, if you want any further information about anything mentioned today, contact me in the first instance, but I will be passing on the details of Melody and Kat, because they have kindly offered to help you do all of your work in relation to MCDA. Okay, thank you very much, everybody. We have now reached time, and I will be stop sharing my screen, and I shall bid you farewell. And I shall see you all again. Thank you very much, and I'll see you all in a couple of weeks. Thank you, and goodbye.
Thank you, guys. Thank you.
Bye.